In this video lecture, we will discuss about the microbial examination of drinking water by the MPN method. What is MPN? MPN is an most probable number or the count of the organisms present in our food sample, drinking water sample or any of the sample. What is the principle or objective for this method or of this method? It is to enumerate the number of bacteria present in the drinking water by the MPN method. To means it is to be count. To enumerate is to count the number of bacteria present in the drinking water by the MPN method. Next, to identify the bacteria present in the drinking water sample. To identify which bacteria is present in that present sample or in the drinking water sample. This is for the objective of the MPN method, which is the first one, or what is the principle, we can say principle or objective of the MPN method is to enumerate the number of bacteria present in the drinking water or to identify the bacteria present in the drinking water sample. What is introduction? In introduction we will see what is most probable number or which type of technique it is. It is an analysis of the statistical method. Statistical method means it bases on the counting or enumeration of the bacteria or the organism based on the random dispersion of microorganism per volume given in a sample. It is a dispersion means how many bacteria are present in a sample per volume. In this method, it is measured the volumes of water is added to the series of a tube containing liquid indicator growth medium. In this method, there will be an indicator medium. Here we will take McConkey broth or brilliant green bile lactose broth. Here, which is an indicator medium which indicate the presence of organisms. The media receiving one or more indicator bacteria shows growth and characteristic or color change. Which type of indication will be seen in that medium? It will indicate the color change and growth of the bacteria. Growth of the bacteria will indicate the color change in that specific medium. The color change is absent in those receiving inoculums of water without indicator bacteria. There will be no color change as the inoculums of water with the indicator bacteria. Those bacteria does not, or the water sample does not cause or does not have any bacteria, will not show the color change. From the number and the distribution of the positive as well as negative reaction, the MPN indicator of organism in the sample may be estimated by this. How much wide the organisms are spread, that can be estimated by the three types of method, which is an presumptive test, confirmed test and completed test. These three types of tests are being performed or followed to identify the bacteria or microorganisms which are present in the water sample. First, presumptive test. What is presumptive test? It is used for the detection as well as the estimation of coliform in the water sample. This test is performed because of specifically for the coliform bacteria. It is used for the detection and estimation of coliforms in the water sample, whether the coliforms are present in the water sample or not. For the estimation of coliform, lactose containing broth medium is used. Here for the coliform bacteria, which is an growth factor, it is an lactose broth which will initiate the growth of bacteria or microorganism present in the water sample. Commonly used medium, it is an McConkey broth that contains an indicator which is an bromocresol brew. The McConkey broth which contains or which have its indicator named as bromocresol blue, which will color change, which the growth indicates the color change from purple to the yellow, which indicates the presence of microorganism in that particular sample. An inverted Durham tube is placed in that tube. Inverted Durham tube is placed in the tube because the microorganisms can also create or form a gas production. The gas production is seen in that Durham tube. The color of the media changes into yellow, and on the collection of gas in Durham's tubes, bacteria are assumed to be coliform. What are the characteristics of coliform if present in the sample? It will color change from purple to yellow, and it also creates or produce the gas in our Durham's tube. The number of positive tubes are counted and referred to the standard chart to find in the MPN of total per 100 ml of the water sample. McCarthy's chart is been referred as per the counting of the number of positive tubes, which has been followed in that presumptive test is how to count the number of positive tubes or per micro per 100 ml. How many bacteria or microorganisms are present per 100 ml of the sample. Next one is an conform test. Conform test, here some spore forming bacteria gives the false positive test in the presumptive test. So we have carried out this further by the conform test. Some spore forming bacteria which will give the false positive test. In this, the conform test is done to determine the coliforms are fecal origin or not. Here it is been confirmed that whether the coliforms are from the fecal origin or not. They are the E. coli or not, or we can say the E. coli are from the fecal origin. There will be the presence of E. coli or not. For the positive presumptive test, the inoculated in the selective media like EMB and incubated at 37 degrees Celsius. If E. coli will be present, it will be checked by inoculating the sample on the EMB agar or eosin methylene blue agar, which will develop and green sheen colonies, which will indicate the presence of E. coli. Presence of typical colonies at 37 degrees Celsius confirm the positive coliform test, and those at the 45 confirms the presence of E. coli. This procedure has been followed for the confirmed test. The next one is an completed test. In this test, the subculture of typical colonies in lactose containing medium. Here those EMB, the growth or bacteria or microorganisms present on the EMB agar is been subcultured in the lactose containing medium and incubated at the 37 degrees Celsius and 44.5 degrees Celsius. The presence of E. coli is confirmed that the production of gas at the 44.5 degrees Celsius. Here the E. coli will be confirmed by production of the gas at the 44.5 degree Celsius. What are the requirements for this practical or for this method? First one is petri dishes, test tubes, sampling bottles, McConkey or lactose broth, EMB agar and nutrient agar, Durham's tubes which is used for the production of gas, test tube strength and the water sample. Here are the procedure for the presumptive test. Prepare a McConkey purple media of a single double strength in a test tubes with the Durham's tube and autoclave it. Take the 3 sets of test tubes containing 5 tubes in each set. 5 tubes is present in the each 3 set. With one set will be containing 10 ml of double stranded medium. Another two will be containing 10 ml of the single strength volume. What is double strength? Means the double of the media, and single strength is single strength of the media. Here in the double strength, the media will be consumed in the double strength or in the doubled amount. Using sterile pipettes, transfer 10 ml of water to the double strand broad tubes and transfer 1 ml of the sample to the each 5 tube containing second set and in the another 5 set containing single strand broth transfer 0.1 ml of inoculum and incubate it at the 37 degree Celsius. After incubation, we can see the production of gas and color change of the media. Record the results. In conform test, what you have to do in conform test? Take a positive tube from the presumptive test using EMB in a duplicate. Incubate those tubes containing positive will be inoculated or it will be streaked on the EMB agar plate. Incubate the plate at 37 degrees Celsius for 24 hours and another plate at the 45 degrees Celsius for 44.5 degrees Celsius for 24 hours and look for the typical colonies in the media. Blue black with the green metallic sheen colonies are of the E. coli in the EMB agar. What we have to confirm whether it is an E. coli or not. E. coli will produce a green metallic sheen on the EMB agar. For the completed test, here we have to inoculate a colony in a tube of lactose broth. From the EMB agar, we have to take a E. coli colony or the colony which we have to inoculate in the lactose broth with the Derham's tube present in the tube. Subculture the colony in the nutrient agar. This subculture is considered to be an option. It is to be an optional. If we have to do, then only we have to for preservation. Incubate that broth cultures at 37 degrees Celsius and at 44.5 degrees Celsius and the nutrient agar at the 37 degrees Celsius. Here the nutrient agar is an optional case. If we have to strict, then only if it is in a optional case. It is not necessary to strict on the nutrient agar plate. On the next day, we have to examine for or we have to note for the acid and grass production in lactose broth. The nutrient agar is used for the gram staining and the IMVIC test purpose. Here nutrient agar is used for the performing the gram staining and as well as to perform the IMVIC test. Here that's all for the MPN. Here the most probable method is being used for the confirmation or to check whether the water sample is contaminated with the fecal type of ori or fecal type of organisms, or we can say the presence of coliforms in the water sample.